There we go. So just if you have any issues with that, just know that we're recording. So good morning. Thank you for joining us. And we really appreciate having Dr. Yao with us. Congratulations to him on being awarded one of the High Path Challenge grants. Really proud to have that acknowledgement of his work from USDA, and appreciate the time and effort he put into applying, and will commit to this project over the next, couple years, to helping us solve the challenge of highly pathogenic avian influenza in Indiana and beyond. Dr. Yao is originally from China. He began his academic career in the United States in the year 2000 at Pennsylvania State University and came to Purdue in 2005. In 2011, he was elevated to Associate Professor of Food Science at Purdue in our College of Agriculture Department of Food Science. His specialty areas include work with starches, and you'll hear more of that in his discussion. You'll see how that kind of has translated into his interest in this proposal here. So without further ado, Dr. Yao, thank you for being with us. The floor is yours.
Thank you so much, Becky, for creating such a wonderful opportunity for us to share some kind of details about this project. And thank you so much for everyone to join our discussion. So very quickly, my background is on carbohydrates. I was hired by Research Center as a starch person, working on starch. And then we transfer our focus from starch to some other carbohydrate-based nanoparticles. So all of the work that we are going to talk about, they are pretty much based on a common platform, which you can call that carbohydrate-based nanotechnology or something like that. So that explains why I'm in the food science, but we are doing drug development work. So now, please allow me to share the slides. I think this is okay. This is the last slide, actually. Okay, so let's start. Let me get this pointer. Can everybody see the slide? Okay. So
okay.
Yeah,
I can.
This is yes.
Okay.
So this is part of the HPAI Poultry Innovation Grant Challenge announced earlier this year, and we applied. We know that over 400 proposals were submitted, and the USDA office, they sponsored about 50, a little bit more than 50 proposals, and we were very fortunate to be one of these proposals that has been funded. So the topic of this project is very straightforward. The keyword, we only have three of course, for control of HPAI, they are very important keywords, but technical keywords, only three: aerosolized, soluble niclosamide. So what I'm going to do is to talk about why niclosamide, why it should be soluble, and why we should fog this soluble niclosamide solution to the chickens, to the birds. So this is like a background introduction. We know it's a big challenge. HPAI is a big issue. Almost 100% of the mortality. There's billions of dollars of losses with our poultry farmers. And current the way is to depopulation. We call this a stamping out, but it's very costly and disruptive. And for vaccines, there's some potential, but it has not been approved due to all kind of reasons, such as drift, label injections, and DAVA issues. And also, there's no human antivirus has been approved to use for birds because there's a lot of concerns in terms of the resistance development of these antiviral drugs. So now there's a gap. we define, according to our proposal's direction, we define the gap to be there's no flock scale inhalable antivirals. So the project, the goal of this project is to develop aerosol of soluble nicotinamide for respiratory protection. Okay. So this is just an overlook. The idea of this is to make a point that the respiratory tract is the point that needs to be protected because this respiratory tract is a primary target. So this is like a chicken, respiratory systems, lungs, air sacs. This is like a schematics for this. So this area is a primary target for the virus. And if you do the intranasal challenge, there's going to be 100% of mortality. Okay, it's very effective. And the virus, they can come in from all kind of paths in air, dust, or formats, or something which is unknown, which is a huge challenge. And but at least we know that most of the time the virus they spread to individual birds through airborne spread in most of the U.S. outbreaks. So that's why we say, okay, if the respiratory system is a point of being attacked, so then let's just focus on the protection of the respiratory systems. So that's why we say, okay, let's do inhalable. Assuming the birds get the virus by inhalable approach, now let's protect the chickens through inhalable pathways. So that was the rationale of aerosols, of fogging, this type of techniques. Okay, let's get into the actual drug. Excuse me. So this is the actual native, the original nicotinamide. It is a yellowish powder. It's very cheap. It's like $30 per kilograms. So this thing is very cost effective. This is a molecule. The only issue here, we're going to talk about this again and again. The only issue of this drug is poorly water soluble. If you throw that in the water, it's just like sand. Really doesn't show any efficacies because the solubility is extremely low. So and also and what we want to do is to repurpose this nicotinamide, which is currently it's a drug to treat the tapeworm. We want to repurpose this drug into antiviral drug. But actually, this is a big area. There were many papers published to activate this drug for antiviral efficacies and also for anti-cancers. There's all kind of publications here. And just for antiviral activity, it's very broad. It has been reported to inhibit the growth of virus with SARS-CoV-1, SARS-CoV-2. SARS-CoV-2 is a virus causing COVID. And influenza A, dengue viruses, Newcastle disease viruses, and even porcine epidemic diarrhea viruses, as well as HIV or some other things, which I didn't really put here. There will be a long list. Dozens of viruses can be addressed by nicotinamide. And also, it's very effective. It has a very low macromolecular inhibition of 50%. So which means it's very efficacious. And there's another very, very important thing is this drug inhibit the virus through changing the environment of the cell, of the hosting cells. So when you apply this drug to the hosting cells, the cell is going to change its internal environment, and this environment is not welcoming to the virus. So it is a host-directive antiviral, so it has a very high resistant barrier. So there's no concern. I should say there's very low concern that you use this nicotinamide a lot, then at a certain day, suddenly you get a resistance for that. This concern is extremely low due to the mechanism of this nicotinamide. And also there's a South Korea company called Hyundai Bioscience. They are developing this nicotinamide-based drug as well as for this HPAS virus. But most of their work is for human because there's all kind of concerns that potentially next pandemic could be this kind of virus jumping from chickens, from dairies to persons. So there's a lot of concern. So there's a lot of preparation to develop new drugs and new vaccines for this. Okay, so this is niclosamide. So this is about the niclosamide efficacies, but this is niclosamide in terms of the environment. So the question is, if we apply all kind of niclosamide, fogging, or this kind of things, how would that affect the environment? The good thing is this compound has been used a lot in the environment. So here it has been approved to be used in the environment. Here's a description. And there is a, I put this part, make it larger so you can actually see it has been used in the US, it has been used as a lamprey site to control sea lamprey larvae and mollusc to control freshwater snails. So this kind of things. And also the study has found that niclosamide is not persistent in the environment. It can break down through hydrolysis, proteolysis, and microbial degradation. So it's a quickly biodegradable compound with minimum impact on the environment. So that's a very good thing for fogging applications. As we just mentioned, niclosamide is kind of like a very ideal, compound or drug for repurposing for anti-cancer, antiviral, or for any other things. But there's a huge barrier, which is niclosamide is poorly water soluble. It cannot be effectively solubilized. So because of this, it has a very low bioavailability. So when you deliver the niclosamide, it just cannot get into the system or cannot solubilize to show its efficacy. So now here is like our novelty. So what I just talked about is the background. It's public knowledge. So our novelty in our group at Purdue is that we have developed a drug formulation where we use OSPP to solubilize niclosamide, to realize the liquid solution, to precisely control the dose, and also because clear liquid, it can be used very effectively for fogging. And now let's talk about OSPP. OSPP is octanyl succinate, hydroxypropyl fetoglycogen. And the core of this compound is fetoglycogen. And on the fetoglycogen, we do two chemical modifications, octanyl succinate and hydroxypropyl. Both of this modification, they are food safe. Has been broadly used for food and also for pharmaceutical areas. So next, let's talk about here the core of fetoglycogen. So in my lab, we have been working on fetoglycogen for many years, for over 10 years, 15 years. Fetoglycogen is a starch type nanoparticle extracted from traditional sweet corn. Okay, so it's all natural. So this is corn. It needs to be a sweet corn, traditional sweet corn, not the super sweet. But now you have this shrunken kernels. From these kernels, you can extract the individual nanoparticles. There's a scale bar, which is 100 nanometer. So every individual particles, they're spherical shape. They are about 50 to 70 nanometers. It's a real nanoparticle. One nanoparticle is one molecule. Okay, so we did some studies on this to understand the structures, branching, all these kind of things. And at the same time, we do a different type of the surface modification to functionalize this nanoparticle. And among all this functional nanoparticle, OSPP is one of them. So this is OSPP. So the core is a nanoparticle. At the surface, we put two functional groups. One is octanyl succinate, which is this part, conjugated to the SP part, and all this chain attaching to the nanoparticle center. So if we magnify this, it will be like this. So you can see the individual glucose unit connected, but the green ones, the hydroxypropyls, and the red things, the octanyl succinate. So these two things come together. They are very functionalized. They're very functional, very effective to stabilize the crystallized niclosamide. It can stabilize that in a amorphous state in the water for a prolonged period of time to give this high solubility. So that's a mechanism for, this kind of like a formulation design or, drug design. And next, so this is what we done. So if you incorporate niclosamide with OHPP, this is very transparent. You can see through this liquid. You can see the background, the strips. Without this, it's cloudy. It's not water soluble. So using OSPP, we can improve the niclosamide solubility by at least over 5,000 times. That fundamentally changes the landscape of niclosamide applications. As we just mentioned, the chemistry was food and pharma safe. And also, we use a third-party CRO to do toxicological studies on OSPP itself. And the result shows there's no mutagenicity, and it's a very high safety profile. So that is OSPP. And after we got this OSPP niclosamide, we studied their oral bioavailability. We found that it can improve the bioavailability for up to nine times with prolonged release. And also a couple of years ago,
was it red?
Yeah.
And after also about 2020 and 2021, we sent this drug to the University of Chicago. They have a level 3 lab. We actually tested this drug with the mice that has been infected by COVID. And we have found that this OSPP niclosamide, when they are soluble, even just at the low dose, like a 10 to 20, they are very effective to substantially reduce the lung viral load of COVID virus in mice. But if this control is regular niclosamide, even at a very high dose, there's no effect against the infected mice. So that was COVID test. And recently, just right before we submit the proposal, I sent this drug to Ohio State University. They also have a level 3 lab, and they tested, it's an in vitro test, they tested the drug efficacy and show its activity against 3 types of, the avian flu virus, including H5N1. So that is the pre-works and preliminary data. So because of this, we expect that because now we have this OSPP niclosamide with high solubility, and also we make the inhalation, this kind of formulation, we can have a very high drug load in the respiratory systems. And at the same time, the total drug use is very low. So we will not really get a very high substantial drug load in the system. So basically, the chicken overall has a very low drug, but its respiratory system has a very high drug dose against the virus attack. And also because of this mechanism of niclosamide, this drug is broad spectrum. It's not just the high pass, it's maybe low pass, or maybe some other kind of virus. I'm not sure I can see all kind of virus, but at least the majority of the virus that would affect the bird through respiratory tract. It will be able to protect the bird from those. And also, as I said, because you deliver the drugs through the respiratory tract, you can accomplish a very high epithelial lining fluid concentration, which is much higher than effective, the drug dose, while still make the drug concentration very low in the system to maintain a high safety profile with the bird. At the same time, we need to make sure that the fogging, the liquid droplets needs to be small enough to be able to penetrate into the lung, into the air sac, to make sure the whole system can be protected. So the droplet particle size needs to be precisely controlled for effective protections. And also we consider that you can fog the chickens we can frog the birds before or after the potential viral infection. If before, that is preventative, we call this a prophylactic effect. After the effect, basically we call this therapeutic. So we are going to test both scenarios, before or after the challenging test. Okay, so that's pretty much the background information. So based on those, this is our general scope of this project. We want to optimize the production of OSPP nicotinamide formulations. We need to generate more, like more than hundreds of grams, maybe kilograms of this scale. Okay, and then we are going to do two things at the same time. First is to develop the fogging systems, and now we do the fogging test of birds to understand the distribution of drug in the respiratory systems and also in the host systems, to understand the pharmacokinetics, to understand if the how the dose and particle size is associated with the distribution of the drug for the effective protection. And at the same time, we're going to do the challenge test. Basically, the Ohio State University, they have a laboratory lab. We give them the drug. They're going to infect the birds with virus, and they're going to apply this drug before or after this infection to understand the protection, the level of protection. So there's a lot of work over there as well. So after both work is done, we are going to do a comprehensive toxicological test. Basically, you apply this drug repetitively for several days, for a week, to the bird, and then you study if the bird is still good, are they still healthy, and as well as the distribution of this drug in the whole systems. And also that's very important, which is related to food safety, which is, the question is, okay, you apply nicotinamide to the birds. How about the nicotinamide content in the muscles or in the eggs? So we need to understand the withdrawal or let's say disappearance of this compound nicotinamide in the eggs, in the birds, to understand that when you're selling this bird or you're selling this eggs to consumers, the amount of the drug is none or is negligible, or at least it should be regulated. So that's the final work for this proposal. Okay, so that's pretty much the technical part. What we would like to communicate and discuss with our poultry farmers is how we can work together. So this is like the overall work. So the HPAI, this program sponsored this HPAI project. Most of the work they happen in the lab. We can, of course, we can do all kinds of tests. We can publish papers, discuss ideas, but we also at the same time want to effectively transfer the knowledge and technology to actual application areas. So we really want to establish the partnership with poultry farmers, with animal drug companies, and also we are assuming we are making progress, which we are kind of, I think we have pretty high confidence that we can develop some good things. We want to commercialize this. So we want to build a team. We will be working with FDA, people about how to convert the science into actual products through all this regulatory procedures. And also we want at a certain point to have the investors to support us to commercialize these formulations to support our poultry farmers. That's pretty much what I want to talk related to the project. Now we can have some discussions. Thank you so much.
Thank you, Dr. Yao. Come back on here. Really appreciate that description, and very interested to hear questions that we have participants on from a couple of different areas and forms of production. just as you and I realize it's the first time you're hearing the information and hearing the description, so it does take a little time to process and think about what it might look like in your flocks and in your production systems.
Yes.
But any thoughts from our participants on potential that you see here, questions that you have, areas you think might be worth exploring first? And you can just take yourself off of mute and ask your question.
So, Becky, this is Vernon, Wabash Valley Produce.
Yes.
This really, I mean, it sounds great, this project, and looks good. I guess, has there been any research or thought, maybe that's down the road, as far as any effect to the bird as far as production?
So I know we talked about withdrawal from the drug, but whether or not like egg production is reduced as a result of administering the drug.
Correct. I mean, and that's probably early, but that might be something to be thinking about, or maybe you have already.
Yeah, I think that's a very important point, and we should incorporate that part in overall research. And also, I think of course, we can do some control studies, look at the performance and behavior of the birds after and before the drug, the fogging. So we should be able to get some preliminary data on that. But eventually, we want to deliver this data to our farmers, and it would be nice that the farmers can also do some like a scale-up studies to understand that.
Vernon, what sort of what sort of things would you want to know? If he's doing a control study, in terms of, I guess, how would you measure it? Is it shell quality and thickness, percent solids? Like what sort of things would you be looking for measurements of?
Well, I'm not really into that side of it as much
Okay.
with our company. Okay. So I just brought it up as a thought process. But as far as an overall application, the fogging, if it works, that's a very nice application that we use already on the farms for flies and things. So that would be a nice vehicle to administer it, though. Okay.
Basically,
maybe Alex
you
is
want
on, or maybe he can comment to the
basically you
production
want to
thing.
make sure that there isn't any egg production loss or feed consumption reduction, that kind of stuff. This is Eric Gingrich, by the way.
Yeah, it's
It
actually,
depends on
I would just say like eggs per hen house could be a good measure.
well, daily
unit of
egg
measurement.
production. If
Yeah.
you apply the drug and they drop 20%, that's not good.
Right.
Or if their feed consumption drops like 3 or 4 pounds. But one of my big questions is how often would you have to apply this? Would it be a daily thing, or how long would one application last as far as antiviral activity?
Yeah, that's a great question. I would say, we have been thinking about this for quite a while. Excuse me. Here's what I would like to say. Apparently, we cannot apply this drug for every day. Potentially, let's say once per week or once per 10 days. In the regular seasons, when you do not see major, risk, and under high risk seasons, you may improve the frequency, maybe twice per week or something like that. But when you hear this, your neighbors or some issues coming out, like right now, now you have to really, improve the frequency maybe once a day. Basically, we propose just a spray like 5 to 10 minutes. We can control the concentration of the fog. 5 to 10 minutes fogging, then it's over. Because we know how how much air, and as well as the fog, and as well as the drug were being held by one bird. We can calculate that
Sure.
kind of precisely. And so after 5 minutes, we see, okay, the drug has already covered have the protection dose. Now let's stop. So I would say even at the highest risk, scenario, once per day for like a week, after it's like the wave is going out. I mean, now you can come back.
Yeah, it's not a problem to daily apply this. We have
Right.
producers right now in northern Indiana that are applying disinfectants to their flocks daily,
Oh,
fogging
wow.
it.
Yeah.
Yeah, to try to, reduce the load in the house if it is there.
Right. Right.
So, but
E-
the cost of it has something to do with that too. How often you can afford to apply it.
Yeah. We actually in the proposal we were trying very hard to estimate the cost. The number is a very preliminary number in terms of the cost of OSPP production.
Uh,
About the naclazimide. Naclazimide is a cheap part. It's very affordable.
mm,
Um, and OSPP, our new solubilizer, is something we need to prepare that. But as well as the spraying system, the fogging systems. From this kind of analysis, we calculate the number. The rough number is if you apply for 7 days for each bird for 7 days, the cost would be less than 1 cent. Because you are not really fogging a lot of drug. It's very effective for the protection. So actual drug use is actually pretty low.
okay. It gives you some idea.
Yeah, give you some idea.
Okay.
Um,
Yeah,
for yeah.
the other thing is, the food safety thing, residues,
Mhm.
that could mess things up real quickly.
Right. So perhaps so that's the why we want to study that in the final stage of the project. So we repetitively we simulate the most frequent scenario, like let's say 7 days, spray the birds every day, and then we stop. Then we monitor the niclosamide concentration in the egg and the muscles, and to see when it's below a certain threshold defined by none. And the here is I think the next thing is when you spray that, even you spray that for 7 days, the total drug you deliver to the chicken is actually very low. The total, even though it's not being withdrawn, it's still very low. And after a couple of days, hopefully, it's will drop to non-detectable or certain threshold. And also, most of the drug will be in the lung area or in the respiratory systems. So hopefully they are not transferred in the egg, and also in the muscles. But this needs to be carefully studied.
Does someone on the call, and if it's not somebody on the call, we may be able to explore it. We can explore it beyond the kind of fogging systems that are already commonly used. So, maybe it's something that's easily adopted with existing
Oh
equipment
yeah.
or systems.
Yeah.
Yeah, I don't.
Yeah, we fog chickens all the time. Pullets for vaccines, and we fog uh, for insects, flies.
Mhm.
So we
Mm,
have sprayers out there. There's a lot of spray technology out there that's
mm.
being used already.
I
That's
guess, is there
very
one
nice.
that,
Yeah.
yeah, is there one that you see predominantly or that people like to
Well,
work
there's
with or?
a big variety.
Okay.
The we've gone away from in cages. We used to use these foggers that on carts, but now we use handheld foggers. There's electric ones, gas powered ones that you can dial it in and get really fine mist. One of
Right.
the one of the best systems is up in Minnesota. It's I forget the name of the company right now, but he has a real sophisticated spray system that, it really gets a real fine droplet. It's expensive, but it's really, really good. There's less expensive ones that work. So
Is it
they
at all common to have like whole house
yes.
systems that are
Well,
automatic? Like,
these are portable sprayers. You just walk down the house and fog the chickens.
right.
Yeah.
Do you ever
They're
see
not
things where they're installed in the barns? It's just kind of
not typically,
not very
no.
common. Okay.
No. There is one company, a disinfectant company, that was putting a system in the house to fog disinfectant once a day. And that kind of system might be something, more, if we had something like this, uh, that comes to fruition, it might be more people would put that in. I don't know.
hmm
But right now, they have not.
It's actually, I was a little bit late. What is the classification of this? Would it be a medication, a vaccine? I'm thinking from an organic
let's
perspective because
let's go to you're talking about the nature of the drug, right?
hmm
So this is a slide showing the nature of the drug. It's FDA approved anselmidic. It's a drug for address a tapeworm. It's a already approved drug, but
for
yes.
FDA, but that wouldn't be approved for organic.
Now, so we want to repurpose this FDA approved drug from tapeworm drug to antiviral drug. And we are going to do this, and we need to apply for antiviral applications again with FDA for the animal drug applications.
Yeah, I doubt it would be applicable for organics.
Yep.
Organic. Oh, you're talking about organic. Okay.
Organic chickens, yeah.
It'd have to be petitioned.
Yeah.
Another interesting piece to understand is the use in the environment. You have a slide from EPA that talks about, yeah,
This one.
I think that's also informative.
So this drug has been used a lot in China. They dump this drug in metric tons, possibly, I think, in lakes, in rivers, to kill the snails. And so because the snail, they host some kind of very bad, kind of creatures, can attack to people, attack the animals. So this has been used for many, many years. And so now the United States is also using this for the similar reasons, as well as for killing this kind of things. So from the EPA perspective, as has been used for many years, there's no concerns has been reported, and also apparently there's no resistance has been developed by the broad use of this kind of niclosamide. So this is give us some kind of a rationale for, I would say, low concern about potentially the fogging, EPA approval, these kind of things.
Ashley, if there are specific things that you would need for a petition, that might be interesting to understand, in terms of just data being collected as he's going through.
Right.
Yeah. So organic is very different. So I think this would probably have to be petitioned to the national list, which is there's a real process on that. The National Organic Standards Board, they're the one that kind of votes on it first and makes the recommendation to the Secretary of Ag, then the Secretary will put it on the list. I don't think this could be an easy way to where you just like would submit it to a certifier for approval, like I would with like say a citric acid or like that's an easy thing to get approved in like on a farm by farm basis. I think this would need to be broadly approved, and that's a like I say, that petition process is pretty lengthy. I've inquired about petitioning a substance before, like if you use there's some consultants out there that do it, and it's like 20 grand to get it on the list, generally. So,
Yeah.
How is niclosamide produced?
How niclosamide is produced.
Yeah.
Majority,
Is it
this is my understanding. The majority of niclosamide they are produced in China, but also
uh-huh.
Brazil, they are also producing niclosamide. So
Is it a
their pricing is very
chemical
low.
process, I guess, or
Yeah, chemical
must
process.
be?
It's artificial ingredients.
Okay. So it's not a mined product or something like that. Okay.
yeah, it would probably be an uphill battle to get that approved in organic.
Mm. So here's my question, in terms of organic, if eventually we say, okay, niclosamide is not detectable in the egg or in the muscle, even though you have used that before in the history, but can you say this is organic because it has no niclosamide residues?
No, that's not how organic works.
Okay. The history is important.
Yeah. Because even like, you could take that same example of like, take antibiotics.
Right.
If you have a withdrawal period, they're still not allowed in organic, even
Okay.
if it's not detected.
Right. So a question to Eric, you just mentioned that currently the spraying system, fogging system is by individual people. They walk around in the poultry house and spray.
Yeah. Yeah, these
How?
sprayers will spray out like 30, 40 feet on one, sometimes they use two people and they go down the middle of the house and spray on either side and
Okay.
just spray over the top of the chickens.
Okay. And how long does it take? I mean, is that taking?
Um, it might take depends on the size of the house, of course. But if you got a 20,000 bird house, it'd probably take you 20 minutes to do the whole house. Go down, spray it going down one way and then spray it when you come back. Pretty
Mm,
slowly, walks pretty slowly. You turn
okay.
the fans off. We have
Mm.
a pretty well-defined procedure for this, for
Uh,
vaccinations or fogging disinfectants or other things.
so the worker needs to have the PPE. They have their mask or this kind of things.
Yeah, a lot of them wear masks. Right.
Right.
Yeah,
For if it's a layers house, I mean, it's several layers, several layers of the
it
of
can be. Yeah, yeah. Aviaries can be, pretty high. Yes, but
okay.
the fog or the spray or fog it'll reach the whole house.
Okay. So basically, you're saying that just by manually spraying, the spraying can be kind of precisely controlled to make sure that all of
Yeah,
the okay.
yeah, yeah. We have we mix up like vaccine. We mix up with certain dosage of vaccines, and
Oh,
then
okay.
they know how much water it's going to take to pretty much, cover the birds.
Okay.
Yeah,
I think that's good because by doing this, at least before you make a decision to say, okay, let's install a whole automatic system. Let's do this kind of manual spraying test first to see the outcomes. Okay.
yeah.
Yeah.
Yeah, the nice thing about these portable sprayers, you can take them to different farms. Of course, you clean them up between farms, but
Oh, okay.
your
Ah,
expense is a lot less.
right,
Usually you don't
right,
have
right.
one sprayer per farm. So
Exactly, exactly. They're
unless it's
professional.
a real big farm, a big complex might have their own, it has a million birds or something.
Okay, okay.
But be nice to have this right now.
Yeah. There's one thing I would like to discuss here is at a certain point, let's say there's a scenario that a couple of birds has died and in the house, and all other birds needs to be depopulated.
Yeah.
At that moment, we say, please keep 20 birds or 100 birds in the individual separate area. Let's flock them using this drug. Let's observe them. Let's see if they're going to recover, or let's see what's going to happen with them. From the online information I got, it says this can be done, without even get your drug approved. This is like a testing or something like that. So
Oh, yeah. Like a safety trial or
yes, safety trial or efficacy
yeah.
trial. Yeah.
Huh. Yeah,
So
when you're dealing with high path AI though, they're going to be depopulated pretty quick.
yeah,
Yeah, that would be an
So you don't
area
have much time to observe them.
we definitely need to work with our Indiana Board of Animal Health on whether, if they would be comfortable with that to begin with.
Right.
And then if
Right.
so, how that would work. And then that would probably
Right.
have to go all the way up to the USDA.
yes,
But, I think part of being an approved, as part of this grand challenge, there might be some, but I would say in general, something that they wouldn't, that would have to be right place, right time, like pretty quick deployment.
right.
But and because we take pretty seriously the seriousness and the zoonotic potential of this disease, this virus. So, but yeah, at some level, you do kind of need that data. So yeah, we could visit as you progress, and we'll see where we are, whether we have active virus still here in the state. We can certainly visit with the Indiana State Board of Animal Health and then whatever groups would be needed at USDA to see if there's an opportunity for a real world trial. So,
Right.
Yeah.
That would be important.
Yeah.
I know one of the steps for vaccines anyway, you have to go through safety trials in the field. And one way you could do this is take a flock at the end of its life, a layer flock, and say treat them, fog them for daily or whatever, and you could measure the amount of product in the tissues and the eggs. You could also monitor the egg production, that kind of stuff. And then if they go to this, be probably a flock that would go to landfill or something. So you wouldn't have to deal with, going to live bird markets or another place.
Yeah, and Dr. Yao,
That'd
I guess, yeah.
be an idea.
So just kind of follow up, when our egg-laying chickens come to the end of their productive lives, they can go a variety of different directions. Some of them get sold into live bird markets where, they go on and they're kind of slaughtered one by one for individual customers. They can go to rendering, which a lot of our birds go to rendering. They can go to landfill. And some of them will go to pet food even, I think.
Some are even composted and go to as fertilizer.
Yeah, there's just all sorts of things. And we have a lot of, we call them spent hens,
They're
being
not
the third.
not very much value to them either.
Okay.
Right. But that is something that it would be very reasonable that we could ask some of our egg laying companies here in Indiana that when they get to the end of lay period, and could they provide, like,
Right.
I don't know if it could be even birds that would be transported to, pardon me, to ASRAC, the animal science farm up north of West Lafayette. There might be able to have some trials and do some studies there
Right.
with some birds that they're willing to redirect because they're very low value. So that's great thinking. And I think that's very doable. So.
Right.
Becky, if you get to that point, reach out.
Thank you.
Yep.
Yeah. Ashley, I wanted to ask, like, if you had in the organic scenario, like kind of is going on right now, right? In Elkhart and LaGrange counties, if you were kind of in an area where you're like, wow, there's a really high risk here, and you had birds that you had really, the eggs you were taking from them are considered to be organic. If you administered this as a preventative, and it had already been demonstrated that it the withdrawal period and all that kind of thing, that there's not an issue with egg quality, could you just market those eggs differently? And then
Yeah.
I'm not sure how long it might I guess the big question would be, like, if you do that, how long then do you have to wait to go back? Would it just be switching out when the next flock comes in? Would you then be able to consider them organic again, or is there some sort of longer withdrawal? Or not? I don't know if withdrawal's right.
Yeah, so
Oops.
there's
We're having a little trouble here.
antibiotics, but
Oh, hold on. Just start again. I wasn't hearing you.
I'm in the middle of Missouri and I don't have the best service. So it's just, this happens a lot. If you administer antibiotics, anything that's prohibited, the flock for the rest of the class is no longer organic. So if you were to this and it was prohibited, the rest of the laying cycle of that bird, she's now a conventional bird. And then if you place pullets again, the next flock, as long as they're grown organically, they'll be organic.
Great. Thank you. I think that's good to understand. Like, in the current scenario, our organic integrator companies up in northern Indiana could make the choice to treat their birds this way. They would then have to market their eggs differently, as long as those birds were still in production. But with the next flock, they'd go back to that organic status.
Yeah.
So thank you. That's really great.
You could do that, but it's hard to do that in certain scenarios. Like if, say we had it and we have flocks up there. We don't really have a conventional pasture market. So we would just, it wouldn't help us. So.
You certainly wouldn't be able to market the eggs or anywhere near as much as you can, as an organic egg.
Well, it's not definitely like, for a lot of people, there's just nowhere to go with them. That, like, if you're pasture, like, there's just no other market besides organic.
Yeah. Okay. Thank you.
How about, I was just curious about related to current duck situation. In the proposal, of course, we focus on chickens, but if possible, we can also look at the scenario with the duck production facilities.
The ducks they don't raise ducks under organic standards, at least here in Indiana. So that's not a barrier.
Okay.
Dr. Yao and I both attended Dr. Greg Fraley's 5-year poultry science chair summary lecture yesterday. And so we were listening and learning lots about ducks. I think Dr. Fraley would be a great partner. He does a lot of research trials there at ASRAC and also on farms, here in Indiana. So that would be a good connection there. Certainly in terms of, Indiana leads the nation in duck production, 60% of the nation's duck. Well, I don't know. After all our losses, our numbers may be dropping, but worldwide, significant duck production across the world. So having something that would be effective for ducks, certainly in terms of market, it would be there. And Indiana would be a great laboratory for that, considering how many duck farms we have. Right. So yeah, I think that's a reasonable area to pursue, or side area. Yeah. The ducks get the virus and then frequently start to recover from it before they're even depopulated, which is one of the most difficult parts of it. But so, I think we kind of maybe already have a good sense of that, maybe really understanding what the prophylactic, benefits would be, because what I think we're learning, we've always thought that ducks were more resistant to high path. And of late, as I've talked with some of the companies up there, they've said, no, really, the science that they've kind of started to hear is that ducks are more susceptible to high path. And they're really, Indiana State Board of Animal Health was sharing with us the other day, they're really thinking the incubation period for the virus is more like 5 days or 6 days in ducks versus the 7 to 14 we're typically talking about in egg-laying chickens. That's the kind of data I've been hearing just through conversations. There might be some opportunity there.
Right.
Yeah. All right. Well, any other questions that come to mind? I'm sure that as time goes on, you might come up with other questions or other things that you recognize, whether it's industry and out in production, and you're just kind of continuing to brainstorm on what you heard and learned today. If there are other things that you think of, oh, this might be a barrier or it might be an opportunity, please reach out. Happy to get you connected with Dr. Yao and his contact information. It's pretty easy to find there on the Purdue website. And we'll continue to work together and have a partnership going forward. But I really appreciate everybody signing on this morning and all the great questions and conversations. Really just get the juices flowing on this. So
thank you so much.
Yeah. Thanks again, Dr. Yao, for taking and seeing this potential connection and solution, and taking the chance on the grant. And I really appreciate, you stepping in and trying to help us out. So
it's a great opportunity, and I would love to work with you, Becky, the association, and also with our hardworking poultry farmers. We will do our best.
Yes. You're right. All right. Any final comments from anybody?
Very interesting project.
Thank you.
Good. Really appreciate everybody's time this morning. We've recorded the session, and so we'll kind of have it there. If you encounter people who are interested in learning more, please have them reach out and we can share the recording. So, all right. Have a great day.
You too. Thank you.
Thanks.
Thanks.
Bye-bye.
Bye-bye.